address where to meet on Tuesday?
It's on the schedule.
Oh, it's on schedule.
It's on the schedule. Pull it up. So the address is,
Oh yeah, it's right there.
on the schedule. And I'm still trying to determine exactly where we're going to go in the field. There's a lot better flows for measuring in the Santa Clara right now than there are in the Santa Paula Creek. So we'll see how it goes. Okay. So I wanted to give a shorter lecture today because we have quite a bit of activity to do in the fields. I was joking with Emily before class that we haven't really embraced field ecology yet and done something truly tedious. Because one of the things I want you to appreciate is field science is often exceptionally tedious. We talk a lot about statistics and replication. Well, replication means you're doing something over and over and over and over again. And the more variation there is in the environment, probably the more replication you need to be able to capture the range and be able to get a good estimate of the true mean of your population. So we're going to be doing a lot of replication for some vegetation transects, as well as we're going to be putting out our invertebrate traps today. But I wanted to give sort of a larger talk about some concepts in long-term monitoring and how do we understand sort of grander scale environmental change. And one of the nice examples of that is Mono Lake and the tufa towers that have been exposed from the diverting of water from inflows into Mono Lake. Because all the tufa towers that we all see and think are beautiful, you wouldn't see if the lake still had all the water in it. So there's sort of that. Okay. How many of you guys have seen this this graph before? Does it seem familiar to anybody? I hope if you're a junior in ESRM, this seems familiar to you. Yeah? So the interesting thing about this curve is its annotations. So this is the CO2 curve from Mauna Loa. They started collecting atmospheric CO2 measurements in, I think it was the late 50s, and have continued to collect that data actually all the way to the present. One of the big challenges with long-term data sets is how in the world do you fund them? Priorities change in our government, in our science funding, and this curve, if sort of different groups hadn't stepped in at different points, wouldn't be here, and we probably wouldn't have as good of a sense that the CO2 is going up and going up substantially and rapidly compared to what has happened in the past. So you can see there's a couple of different periods where NSF or NOAA have chosen that this isn't a part of our priorities anymore. I suspect you're going to see something like that in the present. And so thinking about that when considering long-term monitoring sets, that things outside the science can often influence the availability of information. But this is one direct record that we have of how the environment has changed over a fairly long period of time. But we often want to know things about how has the environment changed over a longer period of time. One of the questions we started out this class with was, what is our baseline? What is the thing that we're comparing it to? And trying to understand what was the environment like at some unspecified point in the past, and if we're doing a restoration project, what is that point in the past that we're trying to restore our community to? And there are a handful of different sources of inference that folks have used, and one of the best and potentially most tedious is pollen records. So you can look, particularly in shallow water bodies, pollen will get deposited in there in layers. And we'll talk a little bit more about layers that are called barbs in lakes. And you can take cores from those and then analyze the pollen record over time. because you know, as long as something hasn't stirred up the bottom in the lake, that a layer of sediment is going to be deposited, and things that are at the bottom are much older than things at the top. So people can go back and look at under the microscope at pollen and see what plants were present in this area over time and how those plant communities changed. And I think some of the readings that I put together for the islands talk about pollen in some of the drainages out there. We've understood quite a lot about our atmospheric chemistry from ice cores from basically the big ice sheets in Antarctica and the Arctic. We can understand, again, a lot about sort of changes in vegetation over time by pack rats, wood rats. They're basically making these nests, and these nests can last for really long periods, and the rats are going out and collecting seeds and fruits and whatever, and bringing them into their middens, and then they're getting preserved there. And again, there's this layering effect. So things at the bottom are likely to be older than things at the top, though. Rats can things around. And then one of the, particularly when we're thinking of environmental science, and I'll give you an example of human records. Human records are sort of inconsistent depending on where you may or may not actually have any human records to go on. Our area, we do have the benefit of fairly long human occupation and some good written records all the way from like the late 1600s. First, I wanted to show this little video. Hopefully it works. No, it didn't work. No, there we go. I don't know how to make it work. I don't know how to make the video work in the thing, so we'll make it work. Nope.
Subaru gonna go hat mics?
Yeah, Subaru's hooking me up, you know? No, this isn't even the wrong one. Stupid YouTube.
Yes. Another Subaru.
There. So this is a recreation of global land cover change. So basically, they're estimating the fraction of some grid cell on the planet that's under natural vegetation over time, going from basically 8,000 years before the present to the present, and looking at changes by humans of the natural vegetation. So this is really the expansion of agriculture through the world, and thinking about how has the world been impacted, how are these vegetation communities been impacted over time. And when we're thinking of like restoration or conservation projects, how does this sort of information fit into that context? And it'll take a little bit longer before much happens in the US.
Did it just recede in North America?
It did. Before the different Native American communities had different levels of agricultural use. And now you can see where they're predicting it to be in the future. No, we don't need to see the human. All right, so back to us. So in this area, so I work on the Santa Clara River, and the Santa Clara River is basically what created the valley that we're all sitting in. People have been very curious. This is one of the few rivers in Southern California. If you've gone south, you've seen all the river channels in LA, Orange County, San Diego County are pretty much cement line channels. There's some exceptions, but all the major river systems have basically been channelized. The Santa Clara River is still fairly natural. There's levees that have been placed in, and some of the vegetation has changed, and there's substantial water diversions. The company that I work for is one of the major diverters off the river. But the San Francisco Estuary Institute wanted to look back at the historical records and see how has the river changed, in what ways has the river changed when we're thinking about the vegetation communities, the distribution of vegetation, the hydrology of the river itself. So they put together this really cool document. If you're curious about the Santa Clara River, you're curious about what's gone on in this area, it's a really great summary of all the changes that the Oxnard Plain has really experienced. They went all the way from Father Crespi in the 1600s up to, I think, about the 60s. And so you can see the development of different communities in this area, different water uses, and how the river itself has moved through the Oxnard Plain over time. I think within the last 200 years, the Santa Clara River right now comes out near Ventura. It used to come out closer to Camarillo. So the river, like this is a big sandy river channel, and depending on what the flows are doing, it can cut a new channel and move to a different location. So anyway, I highly recommend this document. It's a pretty fun read.
What was it? It was
The San Francisco Estuary Institute. If you just type in historical ecology of Santa Clara River, you'll find it immediately. The first one. Yeah.
I was actually curious, since I did have to read this also, what were the long-term effects of the mining and rock bed that were represented?
Substantial. One of the things we'll talk about during next lecture is about hydrology and sediment transport. And basically, if you take a bunch of sediment out of a river, you're creating a nick point in the river. And sediment's going to get transported downstream by a new flow if there's sediment coming down that's eroding away. And so you're going to have that fill in, but that sediment came from somewhere. And so you start getting down cutting. So you'll see a lot of places with high erosion rates start cutting down. And that's basically what happened in the Santa Clara River. So the Freeman diversion, which I think you're alluding to, that the district I work for runs, is a grade control structure. So basically it was built at the former bed elevation of the Santa Clara River, and so it's holding back the sediment on the upstream side. That does mean there's more degradation on the downstream side of it, but upstream of the Freeman diversion, the river channel is basically at its former elevation. So, and I'll talk more about that in the next lecture. Okay. So, again, thinking about indications of environmental changes, and I kind of divide this into two major categories. The physical changes, thinking of Michael and his interests in sediment transport and how the physical world has changed rather than just the biological world. We've made substantial changes regardless of how biology interacts with the world, thinking about hydrology. And again, my example of the LA River, so this is the way the LA River currently looks, and this is the LA River in 1887. You can see there was still a fairly large development in downtown, but the river itself is basically free-flowing and very wide and sandy. So big changes. And then thinking about, okay, we put in a lot of these seawalls, and these seawalls are generally not to the benefit of normal sediment transport processes on the river, on a river or on a bay. They're generally to protect something on the shoreline. And so those can have really dramatic impacts on what's going on, just in terms of physical processes. But I'm going to emphasize more sort of the biological changes and some of the metrics that we can use to assess biological changes. There are definitely a number of ways that you can assess physical changes. And we'll talk a little bit about surveying here at the end of lecture. And that would be one way that you could assess how it's basically the elevation of things has changed, so erosion or deposition. What we'll talk about, and you guys should have read about, community structure and similarity measures, ways of understanding species diversity and what species are actually present. I hope you guys all will go home with an understanding of what ecological succession is and how that can be relevant to a restoration project, for example. We've already talked some about ecosystem functions, and particularly when we're thinking about, from a resource management perspective, how does that translate into the resources that we're interested in managing? And then how have we, or how have other factors changed the structure of an ecosystem and the way that it operates? All right, so let's first talk about community structure. And I just highlighted a couple of definitions from your textbook that I think are important, and you probably have seen show up in things like the CRAM analysis. That'll show up again, I think, again in some of the invertebrate analyses that we'll do. But thinking about, okay, what are the dominant species? Generally, in most ecosystems, plants are going to be your dominant species. And so which one of these are influential in controlling the structure and composition of what's going on? And those dominant species will often affect the physical and chemical processes. So, you know, tall trees are going to impact the wind dynamics of an area, or the amount of fog that might be collected in an area. They can also be releasing chemicals into the environment. So a lot of plants will actually release defensive chemicals that will help space competitors out in the environment. So these dominant plant species can really dramatically impact what an ecosystem looks like. When we talk about stratification, and I think this was definitely part of the CRAM analysis, is basically how are things layered? Is there a lot of complexity in the plant community? So if you only have all kinds of plants at a particular level, you're probably going to have a different group of consumers or other animal species that are going to be using that, versus if you had multiple different levels, different habitats that those microhabitats that those use. I think in your first quiz, you had a question about dispersion, and basically thinking about just how are individuals of a species laid out in the environment. Are they randomly distributed? Are they uniformly distributed, like an agricultural field or a nursery? Or are they clumped? You'll definitely see some things, particularly like in the desert, where you'll have just clumps of vegetation and then bare spaces in between. When I talk about trophic structure, does that mean anything to you? What does it mean?
It means like the flow of energy in the environment and how it starts at the bottom and goes straight to the top.
Yeah, so it's basically the different sources of energy and who are the consumers in those. So what is the primary producer? What falls into the role of a primary producer?
Photosynthesis.
Yeah. So basically, things that are producing, autotrophs, things that are producing energy. So it's generally plants, algae, and then we have these different levels of consumers. What would a primary consumer eat?
Plants.
Yeah. So they're going to be an herbivore. I think detritivores may also fall into the primary consumer. And then secondary consumers are going to be eating primary consumers, and tertiary are going to be potentially eating secondary and primary consumers. So thinking about how does the trophic structure, how many levels do you have in your community, can impact how the distribution of animals are. If you have really strong control by predation, you may have a different distribution of animals, but then that predator goes away, and you may have an explosion at a different level. Yeah.
Is it? I don't think I read it in this, but like whether it's like top-down control or bottom-up control within that structure, like is that?
Yeah,
something
that
we would record.
that's
or would that be,
something.
like more like a note in our.
Generally, that's something you couldn't just like observe
That
and
might
something
be the question we would be asking.
you'd want to ask, or something that you could experimentally test.
Okay.
Generally, that's been one of the big debates in ecology over time, is how are these dynamics controlled? Like, is it predation from the top, or is it production from the bottom, or does it depend on how many levels of consumers that you have? So there's something called a trophic cascade, and that basically depends on if you have a certain number of levels in your trophic structure, you may be able to have control. But if you have, I can't remember if it's an otter even, then you don't have control.
Okay.
So the other thing that I've tried to hit home repeatedly is thinking about the temporal distribution of animals, whether it be seasonal changes or daily changes in either the composition of the species that you might find or the activity of those animals, because all of those things could be changing and could be impacted by whatever processes you're interested in. You always hear about species diversity in the news, and I wanted to sort of highlight some of the biological understandings of diversity. And generally, when we're going out and measuring something, we're often talking about alpha diversity. We're talking about the local diversity we observed in a particular habitat or within a particular community. There's also beta and gamma diversity, and beta is basically the change in species between adjacent populations within a region. And then gamma diversity is basically taking those on the whole and combining them into a regional diversity measure. I think you guys had to calculate the Shannon index in one of the earlier quizzes, and basically trying to take the idea of not just a count of species, but the abundance of those species should give you some indication on how diverse a population or a community is. And so if I said we had 100 species and all of them had 10 individuals, or if I instead said there was 100 species and 2 of the species had 100 individuals and the rest of them had 1, which of those communities would be considered more diverse by a Shannon index?
One had 100.
100 species and 10 individuals in each one versus 100 species and 100 individuals in the first 3 species and then just 1 or 2 in the other species. Yeah. So where you have numbers distributed more evenly amongst all of them, that would be considered by a Shannon index to be a more diverse community. You can take the Shannon index and then translate it into an evenness measure, where you're basically saying what is the maximum you would expect to have in terms of the distribution of species among, or of individuals among the different species, and then what is our measured Shannon index, and that gives you a sense of how abundant or how even the species are amongst those, and allows you to compare among different populations. You probably saw in the Brouwer chapter that there were like 500 different measurements or different indices that people have used over time, and they may all have an appropriate use at different times. But I wanted to sort of simplify it down to you'll probably see the Shannon index show up somewhere. It's very commonly used, and this is a logical progression from the Shannon index.
What's that Greek letter? Like a Y?
Gamma. Okay. One of the concepts that you guys read about maybe last week was about those indices and how do you compare what's going on over time using a change in an index. And we can use the Shannon index to look at how our species, how diverse is a community and how is that diversity changing over time. But often people are really interested in some particular species. And on the grand scale, there's the assumption that diversity on the whole will also mean that you have the presence of those indicator species, but it may not. And one of the things that the rapid analysis papers that you guys read said is actually in a lot of cases say diversity in plants doesn't then translate into diversity in other species. And so thinking about what are we measuring, how are we incorporating all those things, and then what is our question? What are we really interested in? But I wanted to give some examples of, so we've already seen keystone species, and we had the great example of Pyzaster controlling what was going on with mussels in the intertidal. There's also the example of sea otters and urchin barrens on the California coast, actually all the way up to Alaska, where when sea otters are present, urchins basically are controlled and you have big kelp forests. But when the sea otters started to go away, the urchins took off and you basically have just places with just urchins and no kelp. And then an interesting change happened when orcas appeared to be predating on sea otters up in Alaska, and you started having those urchin barrens showing up again. Indicated species. So you probably all have heard about the least-bells vireo. Well, least-bells vireos don't necessarily, they have similar habitat requirements to the yellow warbler. And so yellow warblers are often surveyed when people are also interested in least-bells vireo to see if the habitat is potentially appropriate for least-bells vireo. So we currently perform riparian bird surveys adjacent to the Freeman diversion, where we're looking at the abundance of yellow chats, yellow warblers, as well as least-bells vireo. If we have a good sense that these guys are really abundant, then it's probably a good habitat for least-bells vireo, and there may be some other reason why they aren't showing up. But if you didn't see any of these guys, then you probably aren't going to see any of these guys either. So ecological succession. So there's the big concept that I want you to get out of this is primary succession. And what does primary succession require? What do you think, Rob? Do you remember in the meeting? Yeah. Does anybody remember?
Yeah.
So
it has to be. There's never, ever, ever plant life there before. So like after
yeah. So primary succession is what's happening on bare rock. So think about like a lava flow. There was no biology there. It was just lava, and then you start having these pioneer species show up, and your community starts changing. So often you start having lichens show up. They start breaking down the rocks, start developing a soil layer. And then you can have other species start showing up. There, primary succession is something that generally only happens when you have sort of massive disaster. Usually you have secondary succession occurring, and so it's starting somewhere more in this area. So there's already some biology present, so maybe there's a soil layer, and then you can start having the succession of later communities. One of the things that I really like about this example and why I'm talking about it in this field methods class is when you're thinking of a restoration, you need to sort of think of the continuum along which a community might develop. And so one of the good examples of this is old fields on the East Coast. So basically the entire East Coast is clear cut during early settlement. But now the East Coast is mostly forested, and you had basically this succession, not primary succession, but secondary succession going on, where you had an old field, and then you start having some of these woody species start taking over, and then the woody species start outcompeting or outshading some of the other species. But then you have maybe a storm event that tears down a bunch of trees, and you could have a meadow open up in a forest. You can also have succession go on where, say, a pond that previously had water fills in with sediment and now becomes a meadow. So thinking about that in the context of restoration projects, or in what you might expect, basically nature changes. That there may be a climax community, but there also may be multiple climax communities. Okay. So now we're going to shift gears a little bit. So you had some quiz questions about these very terms, and sort of going back to what are the things when you go out in the field do you measure? And these are some of the metrics that are really important to understand. Biomass is just the weight of the living organisms that are out there. When we talk about standing crops, we're now taking this and putting it into some sort of known area or volume. So if I just said, okay, well, the biomass of animals that I collected is X, you don't have the context to be able to compare it to somewhere else, because you don't know, okay, well, he went and collected 500 fish, but I don't know how big of an area he collected to collect those 500 fish. So standing crops are one way that you can compare between samples. Production is what is being produced over a given time period. So you're often thinking about that sort of like, say, plants. So you plant a bunch of plants into your field. You know when you started, you know when you collected them, how much production occurred. If we divide that production by how long it was, then we get how much grew per unit time. And so that's a measure of productivity. And we'll talk, I guess later in this one, about gross primary production and ways that people are measuring that sort of indirectly. But gross primary production is how much organic matter is being photosynthesized by our plants, basically, in our ecosystem. The net, so one thing to remember is, okay, so plants are producing by photosynthesis, but they're also respiring. So it's really the net of how much biomass do you make and how much do you consume, and that's our net primary production. So how much biomass is being lost from the amount that's being produced. And then secondary production is how do you translate that primary production, so all of our vegetation, into somebody that's feeding on it. And so the measure that we're going to go and do today, if we had more time in this class, we probably would have taken it to the next level. But so we're going to go out and we're going to put out transects, and we're going to put out quadrats, and we're going to sample the amount of area covered and the amount of stems per unit or per area. If you wanted to convert that to a biomass, you could collect those. And I think, is my next one? Yeah. We could then measure the biomass. What's the problem with wet weight?
It varies how much moisture there is inside, so you can't really compare between species.
Can't be, or even within a species, if like different plants, if one's water stressed and one's not water stressed, if you measure them both in terms of their weight, a lot of the tissue is actually containing water, so wet weight isn't necessarily the greatest way. However, if you say wanted to keep an animal alive, you probably would want to measure a wet weight. Like fish don't really appreciate being completely desiccated before you measure them. So depending on what you're interested in, a wet weight may be an appropriate measure. But in things where you can collect it and you can dry it out, then a dry weight is something that could give you a more comparable answer amongst things. And so that's why we've got drying oven present here. So what is AFDM, ash-free dry mass?
You burn the biomass and then take the measurement after.
Yeah, so that's why we have a muffle furnace. So if you have something with basically a shell or something that has sediment in it, or just tissue that's not biological, ash-free dry mass is going to give you a good comparison amongst your values because you're really just comparing the biology, what was basically burned off at 500 degrees C. When you're thinking about things like sediments, and so people often will use ash-free dry mass to measure organic content in sediments, there's weird geology that also is occurring, and basically clays will have water retained in them, and so you often have to actually take your sample, dry it, ash it, re-wet it to rehydrate those clays, and then dry it again, and then the clays will be re-wetted. And so that's something that really only comes in when you're talking about sediments, but it's the clay's hydration level is something that you want to be keeping into it. We've talked quite a bit, I think, about regressions, and one thing that you can do if you want to indirectly measure things is say if you know there's a good correlation between the length of an animal and the weight of an animal, then you could just use your regression and just go measure lengths of your individuals. Depending on the species, somebody may already have developed a regression, and you may just have to get a coefficient for it. For fish, there are particular regressions that are considered to be appropriate, depending on basically the width of a fish. But length weight curves are really useful for a quicker way of measuring a lot of individuals. But you can also use this sort of form of indirect measurement for measuring other things. So an example that we've used in my lab before is say we want to measure the surface of a rock. We're looking at algal abundance on a cobble in a creek. Well, cobbles are really complicated. They have a lot of different nooks and crannies. And how do you actually measure that area? One method that we have used is basically put a scale bar down and take a picture of it. But that's still only kind of that's going to give you a good estimate, but it's not going to give you a truly accurate estimate. So another way that you can do it is say, take that rock and wrap it in tinfoil, weigh the tinfoil, and you can cut a bunch of pieces of tinfoil of known areas, weigh them, and make a regression that says, okay, for this weight of aluminum foil, I get this area. And that then it gives you sample per unit area. And you can do that with anything that has something consistent about it, paper or foil or whatever you can use to indirectly measure something else. So that's a really useful tool. Okay. So now we're going to shift quite a bit to long-term monitoring. And I wanted to highlight long-term monitoring partially because there's a bunch of different programs, and sort of as scientists but also as citizens, some of your tax money is going to support some of these long-term research programs. I think they're very valuable. You guys read 2 documents today talking about the use and challenges of developing long-term monitoring programs. We had an example of the Hubbard Brook Forest and being able to understand large-scale ecological changes. But there's a couple of different programs that are currently out there. The LTER, Long-Term Ecological Research Network. There's one here locally, Santa Barbara coast, and they're looking basically at the kelp forest and coastal dynamics. So also for somebody who's maybe interested in a career in ecology or in environmental science, these LTERs are often hiring people to help them with field projects. They actually, I think they had a handful of REU, research experience for undergrads, this summer. And I sent you guys out an REU opportunity. It may or may not be interesting to you, but mostly I sent it to you because maybe it seems like it's short term before the next semester goes, and it could be cool opportunity. Paid pretty well. But those programs can be really useful for getting experience, but also they are data resources. If you then want to say something about, okay, I want to understand a kelp forest, a lot of those data are available to you if you have a study that's maybe related to kelp forest. There have been a couple of other longer term projects that were mentioned in one of your papers, like the North American Breeding Bird Survey, an international bottom trawl survey, and those have been going on for, I think, at least 100 years. So you have potentially good information on larger scale dynamics that wouldn't be possible without some of these long-term monitoring stations. One of the other cool things that have come out of some of these LTER sites is trying to understand changes in our atmosphere, changes in primary production over time. And so they put out these things called eddy flux towers, and they're basically measuring a number of different components of the atmosphere. And so they're looking at precipitation, they're looking at transpiration, they're looking at evaporation, discharge from a watershed, gross primary production, and respiration coming out of that system. And they're doing it over a bunch of different types of ecosystems, trying to get a sense of how do we parameterize our models of climate? How do we parameterize our models of primary production? How do we understand those things without going out and chopping down all the trees? Can you measure gross primary production indirectly through these methods? And so that's a really cool thing that's been going on. I think it's been going on for about 20 years. One of the things that Dr. Don Rodriguez is really excited about is citizen science. And from sort of a conceptual standpoint, citizen science has been around forever. Like some of the early naturalists were basically just gentlemen naturalists who were doing this for fun, where we're taking really cool notes and observations about the world. On the sort of more modern scale, when we think of citizen science, it's using the community or using individuals in the community to collect diverse data. And one of the things that you'll probably encounter over the course of your career is it's often hard to get people to go in the field. Like that sounds like all of you guys are really excited about going in the field, but sometimes it's really hard to find somebody who's going to be reliable to go do something, or to find enough people, or to be able to pay people to go and do something. And citizen science is a good way to be able to get a lot of bodies to go and do some particular project that you couldn't necessarily do even with a lot of money on your own. So some examples of these are things like bio-blitzes, where basically you get a bunch of people together, you have a couple of trained individuals, and you send people out to go categorize and catalog biodiversity in a particular area. And so you can look for changes over time. And there is a group, an organized group, citizenscience.org, that is they have an annual meeting talking about the use of citizen science, and have a bunch of different projects that you could look into that people have been using. One great example of citizen science is the Audubon Christmas bird count. Birders are sort of notorious for really liking to make lists of things, and for at least 100 years, Audubon's been getting people out to a number of locations throughout the world, counting birds in a particular method on Christmas Day, and so they can look at changes in species composition or abundance over time. But the challenges. There are substantial challenges with citizen science. And one of those is that not every project is actually suitable to have a volunteer help. Maybe it needs a complicated method. It needs, so say something like tracking may require really intense knowledge of what's going on. Otherwise, you may introduce bias into your information. You may have people talking about some bird that really has never been detected there and probably wasn't detected by that person because they misidentified it. And having done this, I worked as an undergraduate as the exotic plant manager for the south end of the Grand Canyon, and we did this cool outreach project where we said, hey, if you want to help the park, come and we'll do non-native vegetation removal. Well, it turns out most of the non-native vegetation in the park is not in the beautiful parts of the park. It's in like the parking lot areas, the residential areas. People wanted to help the park, and so you bring them out and they go and weed for a couple hours. Well, people sort of lose steam, particularly when they've come to the park with their family. They want to see this beautiful place, and you take them to the Rangers neighborhood and you have them pull weeds. So thinking about, how do you fit that into the context, and what can you really expect out of people who are doing it sort of out of the goodness of their hearts? One component that I haven't really thought about, but depending on sort of the benefits that are ascribed to participation in that citizen science, people may just lie. And some people just lie because they like to lie. We had a staff person who was making stuff up at one point in time. But people may lie because they want to have a higher bird count than somebody else, or maybe they think they won't get any reward if they don't provide some piece of information. So you just have to keep those things in consideration when you're developing a citizen science study. That being said, it can be really powerful, and I encourage you guys to look for opportunities to either participate or lead citizen science sort of projects.
Does that differ much from like surveying the public?
Yes, in that citizen science is actually asking them to go and do science. Like having somebody participate in a survey, that's social science, but in a different way than these sorts of data collection efforts that people are looking for. I'm not a social scientist, so I can't really speak to the survey aspect of things. But I could probably find you a paper. One of my colleagues worked on, so creel surveys are a big thing.
Creel?
Creel, so like people go fishing and you ask somebody to report, how many fish did you catch? And that's an indirect way that you could say measure how a stock is doing. So you are interested, somebody's been stocking a bunch of trout in a creek and you want to see how many fish people are catching and how does that tie into what you think the population is doing in the creek. And maybe you directly sample it as well. But he was able to look at how do you design a creel survey and how do you interpret creel survey data to then translate that back into management level of information. But if you remind me, I could probably find that. Okay, so wanted to put up a cool picture of Death Valley to make a big shift. So we're going to stop talking about sort of conceptual things, and we're going to talk more practically about some methods that are more related to spatial data. Okay. So I know, how many of you guys, could you remind me how many of you guys have done a GIS class? So most people are at least vaguely familiar with GIS. I want to talk a little bit about GIS and some of the things that I think are really important to remember, because if you don't, your data are much less valuable. And one thing to very much consider is when you're taking, say, a GPS point, your GPS point will tell you what is the datum that it is using as its reference. And a datum is just, by definition, is just a fixed point that is the start of your scale. So what is the 0, 0 of your coordinate system? And we're going to use datums a little bit later in terms of surveying. And it's, again, it's just like, what is your point of reference? What are you comparing these to? When we talk about GIS data in particular, we need to think about projections and coordinate systems and how those two fit together. Because if you don't consider that, then you may end up giving somebody a piece of information that then puts, the point that you took in Cam Park somewhere out in the middle of the ocean. And that obviously isn't very useful to them or to you. So projections are a way to represent the three-dimensional surface of the Earth in a two-dimensional plane. When we think about like Google Earth in its original days, they did do a projection. They just flattened out the Earth, but now they're displaying it as a globe. But most of these projection methods are treating the Earth as a sphere, which isn't really accurate because the Earth isn't actually completely spherical. It's more of an ellipsoid, and it's got weird lumps and other things going on. Or they treat it as cylindrical, and again, that's really just like basically taking the world and trying to unwrap it and make it flat. So then you can make a map, you can make measurements, or you can make comparisons among things. When you're using Google Earth, I believe it uses a Mercator with a WGS84 datum. The other component that you need to think about is your coordinate system. And the coordinate system is including a projection. It's a reference system used to represent the locations within some sort of common framework. You can use different sorts of coordinate systems, and that matters when you're actually trying to do a measurement in a GIS program. Like, how is it being treated? What is the coordinate system referring to when it's making measurements? And generally, a coordinate system, and always a coordinate system, includes what its unit of measurement is, and it's typically either feet or meters. When you're thinking about things in lat long, it's going to be in degrees. Your coordinate system is going to include a definition of the projection that it's using, basically to take that 3D information and make it 2D. You'll often see things like defining the central meridian, so basically the line in the middle of that reference, or for different reasons, people will shift from the datum to another location. I don't exactly know why people are doing that, but I know it exists, and I know that it can really mess up your information. So typically, GIS systems can use these different coordinate systems and projections as necessary. It's gotten a lot better over time, and more programs are inherently including projection information as metadata in the spatial data sets that you're putting together. But there's a lot of legacy data out there that doesn't necessarily have that tightly tied in, and so you really need to think about when you're collecting data, do you specify? You'll often see, say you give information to State Fish and Game, they're going to ask you what's your datum, what's your projection, what coordinate system are you using, so they can then take that and put it into GIS. Because if they don't have that information, the spatial data is not really in use. Okay, so that's the message, and I would definitely suggest either taking a GIS class or doing some reading on GIS. Even if you are never going to use GIS, you're going to ask somebody else probably. If you stay in this field and you do anything with the environment, you're probably going to ask somebody else to do GIS for you. And understanding what are the limitations of this sort of methodology, and what are the needs that they have, what do you need to specify to them so they can do their job. And I want to make sure that we're all clear under the conception that GIS is not just making maps. Making maps is really cartography, and making maps is very useful, and you often will need a map associated with your project for some reason. But GIS is a lot more than that. It's a method of storing your data in a spatial context. You can do a bunch of analyses with data in a spatial context. And so you can say, okay, we have a bunch of point data. I can ask, well, how close are those points to something else that I'm interested in? One of the things you'll often have to do, say you're laying out a project area, somebody's going to say, oh, could you put a buffer around it? So you guys made buffers when you were doing CRAM. Buffer, if you say had a transect and you wanted to buffer that area, you could say, okay, make a line and then put a width of 20 feet on it, and then you would have that in your GIS system. And then you could do analyses like proximity analysis and say, okay, I have a bunch of data points in my spatial context, I did a transect, and I want to say how many of those spatial data points were within 20 feet of my line. I can do a buffer and then analyze my information to choose out those. You can look for clusters in your data, so how are things connected in the world? Basically, when you're using Google Maps for navigation, it's basically doing these analyses and looking at connections on road networks and assessing, well, what's the fastest route to get to some particular thing. Sometimes you may need that for biological context, and there are JS frameworks that allow you to do that, or to estimate densities, or look for clusters of things with, say, similar parameters. You can have multiple layers of information within a GIS system, and you can use that to analyze different things. So say you have a layer of vegetation, and you have a layer of invertebrate data, you could compare how do my invertebrate information compare to the vegetation information that I have. And people often use these to do spatially explicit modeling. So some of the things that you guys saw 1 or 2 lectures ago, where I showed you the climate envelope modeling, that was all done in GIS. Somebody had climate information for a grid of cells, and they said, okay, let's try and use that to model the distribution of species. I wanted to do a little practical sidebar on raster versus vector data. Does that mean anything to anybody?
Raster is cells, basically. Like a picture is
Mhm.
a raster because each cell has a different color. Vector is points line for polygons where they have attributes and you can kind of overlay each other also. And
Mostly, because we can most simply define a vector is basically it's a mathematical equation describing a line or a series of lines. You can have, with pixels, you can have data associated with a particular pixel. When you're thinking of, this is a very practical sidebar, when you're thinking of making figures and exporting them to a picture type, if you had a graph with a bunch of lines in it, what sort of file would you maybe want to send to somebody? Can you think of different file formats that might more appropriately share that? Say, I want to be able to scale. I want to give you a figure that's about this big, and you maybe want to blow it up onto a poster.
Vector.
Yeah, so you would want to get that data as a vector because it's a mathematical equation, so it's basically just going to do some multiplication and make it much bigger. The resolution is going to stay the same. One thing to consider, though, with vector data in the context of GIS is what's the precision of the information. So how many little nooks and crannies are included in the actual measurement of that for that equation. When we're thinking of rasters, we're thinking of cell sizes. Yeah.
Would vector be appropriate for environmental data? I mean, nature's not uniform, so vector seems like it'd be more of a uniform concept. Am I right in that assumption? Or what do you mean by environmental?
Yeah.
Like trying to figure out the density of X plant, like they could be in certain areas. Like, would you do a vector format of those areas?
So vector versus, at least when I in photo editing stuff.
Mhm.
So when you're doing it, if you draw a polygon on your map, if you draw it as a vector, it's any shape, regardless of how complicated it is. Especially if you're drawing a border around a tree, even if it's all crazy, or a coastline. Especially if you're doing it in vector, it'll come up with a mathematical equation for that shape you drew. So say, he's saying, if you want to make it bigger all of a sudden, you want to make that image bigger, rather than it just having points in these different cells where the shape is, and then you change the size of that cell, it's gonna it's like Microsoft Paint when you make something,
When you
make
blow
it
something
bigger,
up.
and then it's all weird looking. But if you do it in Photoshop in a vector, where you're using math to define those lines you're making, to define your image, then when you change that image, it's not going to have any distortion.
However, sometimes, so you're taking a bunch of photos with your drone, right?
Yeah.
You're getting raster information out of that. You may want to translate that raster information into vector information, depending on what you're trying to do. So if I were interested in, say, the borders of a river, I probably wouldn't want a raster of the borders of my river. I would want vector lines to describe the borders of the river. But if I wanted to, say, describe the amount of green, so if I wanted to do like a vegetation analysis and I said, okay, well, cells that are more similar to this color of yellow are going to be this kind of plant, cells that are this color of green, or this kind of plant, I'd probably want a raster, and I could do an analysis of how many pixels of those different types were present in my data set. So they're both appropriate, just depending on the context. And when we think about it in terms of what Daniel was talking about in terms of producing like figures for something, I generally work in raster in vectors until I can't, and then you export it to some sort of image file. I would generally recommend for things like graphs that you probably are going to do it in like a PNG or a GIF, because those are specifying particular pixels and the color of those pixels. If you wanted to send somebody a photo, you would probably send it as a JPEG, which is a compressed format. It has interpolation between the different cells. And so you don't get very clear lines. So you could do a simple experiment, like make a graph, export it as a JPEG, export it as a PNG, and see when you zoom in on it, how clear do those lines stay? And it's going to be much more clear. But if you need to do something that has lots of colors and lots of complexity, something with a compressed format that's interpolating between points is going to be better, and it's going to look better when you present it. So, okay, so let's continue on with the practicality. So I wanted to talk about surveying. I'm not sure whether we'll have time to do surveying in the field today, but generally you're trying to get some sort of precision information about a location of interest, and you typically are trying to get it in 3 dimensions. You can use, it's used for a lot of different applications. Say you're trying to measure the slope of something. You want to know, say, you want to know how steep is a river channel. You could go out and measure the slope of that. And again, remember, everything is going to be relative to whatever the datum is that you specify. In construction or in some ecological studies, maybe you need to have a line that's level. You can use a surveying technique to have that line be actually level over a long space. People are often using it to assign like the boundaries of their plots. Like in very practical terms, it's used to like determine the borders of your property if you own land. A lot of ecologists and geomorphologists are often using it to parameterize a model. So we'll go and collect some stream channel cross sections. You guys already did an informal stream channel cross section in CRAM, but we're going to actually do one where we're actually measuring accurately the stream channel cross section. And I thought it'd be kind of cool for you to be able to compare how good were you at sort of sketching in a cross section versus actually going out and measuring the cross section. But people can use this in programs like HECRAS, which is produced by Army Corps of Engineers, looking at how does river channel interact with different flow levels. And if you have a bathymetry, they can do modeling that assesses, okay, well, what are the velocities going to be in the stream, and what locations are those velocities going to be? Where do we expect like a bridge is going to have a problem? So you can do this sort of modeling. There's really kind of 5 different techniques, but I'll just talk about 4 of them. The basic idea with a lot of surveying is you're trying to do a triangulation. So if you have, a point from here, and you have some sort of datum reference, so you know that this is going to be 0 degrees and this is going to be this. You can survey to a number of other points and measure the angles of these. Go to another location, survey the angles to all these, and you can figure out from that information, basically what a GPS is doing is is triangulation. It's looking at the angles between those and then figure out where those intersect, where those lines intersect, and then you can identify the location of that by trigonometry. Trilateration is something that you would be doing with what I'll talk about a little bit later, a total station, where you're actually measuring the distance between these points as well as the angles. So it means you don't have to go to quite so many locations to parameterize a particular location, because you know this location, the angle, and the distance between them, so it's more easy to specify exactly where it is. A traverse is basically just a transect. It can be, a traverse could be a loop, which is a closed traverse, or it could be open, so you could just be going, and it doesn't have to be a straight line, it can be just in some particular direction. And leveling, which is the kind of surveying that we're going to do, is basically used for measuring an elevation of something. And you generally would use an elevation that's known, some sort of datum that's known. There may be a monument somewhere in Camp Park, but I don't know. Or we may be able to get what the elevation of like the bridge is or something. I don't know.
We took some points out there, but we haven't gotten that data yet, so eventually we'll get it.
Cool. So there's a number of different techniques for surveying. I'm going to talk about the sort of physical on-the-ground techniques, which is the one we're going to use this guy at the auto level. And the auto level is a very simple instrument. It's basically just a telescope with some crosshairs in it. It's got a little bit of a zoom to it, and it has some ability to accurately level it. And then inside here, there's this like a hanging prism that if you're not quite level, it actually falls and hangs level, and so your line of sight is actually level regardless of whether you have it a little bit pompous or not. And so you're using this to measure typically just elevations. A theodolite is an optical instrument, so this is sort of an older style where you can very precisely measure angles in this direction and angles in this direction. The modern equivalent, and these will often also be called theodolites, is a total station, but the total station then has electronic distance measurement within it, so you'll use a prism and a laser that's inside this to actually do measurements to your particular points. When you're using these, they actually use like these chains that you stretch between locations so that it doesn't stretch or anything, and so you can measure the distance to things. And so you'd be using this one for trilateration, you'd be using this one for, what do they call it? For leveling. Okay, I wanted to give you a cool example because this was one of the coolest science experiences of my life. I was working in Alaska for a summer in undergrad, and I got to fly to the Bagley ice field, which is in southeast Alaska in the Chugach Mountains. And we went to a place called Iceberg Lake in Wrangell-St. Elias National Park, and I was working with basically a geologist who was really interested in what's the history of this lake over time. So there are a number of lakes that are dammed by glaciers that do something called Yokalhaup, where they basically will drain underneath the glacier all at once. And they'll do it sometime every year, sometimes every couple years, sometimes very regularly, and sometimes unpredictably. And he had observed at this location, and I have a picture a little bit later, that it had multiple stable shorelines over time. So it had filled up to a particular level when the glacier was at some particular height, and then it would drain from that elevation. And then at a later point in time, it came down and it had a different stable shoreline. So we were supposed to go out and drive around in the Zodiac and measure the bathymetry of the lake, but 3 days before we went out, the entire lake drained, and so we ended up getting to walk along the bottom of this glacial lake and basically survey it. So one of the things I wanted to point out to you, and some of the GIS guys have probably already learned about it, is basically correcting your GIS data. And so we had a fixed base station here, so you know this guy isn't moving. its GPS location may say it's moving. And we had a portable unit that we could go and do our survey locations with, but we know this one isn't moving over time, but even though the GPS point is moving, you can use that information to correct the shift in the one that you are actually moving around. I will say that GPSs have gotten so much better than they were in 2003 or 2002. And one of the pieces of information that we were collecting, we were trying to understand the history of this lake. And so there's me digging into the side of a carved out, so the river had carved out through the bed when the lake drained, and you get these really cool things called barbs, sediment layers. So during the wintertime, flows coming into the lake, and again, we'll talk about it more in the stream and sediment transport stuff. Velocity matters. High velocities mean you can transport bigger particles. Summer high flows, because things are melting, you transport larger particles, so you get sands during the summertime. During the winter, there's still some melt going on, and you get these fine clays, and so you get these really nice layers, and you can see it in that figure over time. And so he was able to collect cores of this and go and basically count the layers, and we were also able to extract organic material from this, and then he was able to do radiocarbon dating to look back at the history. In this picture, you can kind of see one of the stable shorelines right here. This is the current shoreline of the lake. I think there's maybe another shoreline here. Over time, he was interested in trying to understand the history of that. So he also was using lichenometry. So different lichens, so remember our concept of primary succession, different lichens show up at different times, and different lichens grow at different rates. So he could use the lichens to assess when was the last time this was disturbed, and if he collected enough pieces of information about that, he could understand when was this underwater and when was this underwater. And that's Mike Loso standing underneath this awesome grounded out iceberg. So, and the whole time we were out there, all the icebergs were just breaking apart into just these massive crashes.
Did you see the Industrial Revolution?
Say again?
Did you see the Industrial Revolution within the ice pack?
I don't know. I actually, so I found that picture of me last night. I was like, oh, that'd be a cool example for surveying. And I typed in his name and I found this NPS publication. And I'm reading through it, and I was like, oh, he did actually publish this. And then I look at the front, and like, the thing says Mike Booth. And I'm like, oh, that was me. That's me in that picture. I thought it was him. And anyway, I was kind of tickled because I didn't know that there was that picture out there of me, and it was cool to see that story has actually gotten out there. So GPSs have gotten much better, and now we have something called RTK, which is real-time kinematic, and it's basically doing the corrections on the fly. When Mike and I were out collecting data on Iceberg Lake, he had to download the data from the base station, download the data from that, and then go back and post-correct it. And Trimble at the time charged inordinate amounts of money for their post-correction software. Now you can just pay an inordinate amount of money to get something that does it on the fly. And so typically you're still going to have a base station, whether it's a base station that you're paying for access to, so there's often navigational base stations around, or where you actually have your own base station with you. And you generally would locate that on some sort of known benchmarks, so you know what the elevation and the location are, and then you can use it in combination with your GPS unit. And people are tying these sorts of things into GPS or into drones, and so it's very powerful to get surveying information fairly quickly. Some of the other things that I know the drone folks have been working on is photogrammetric processing. So basically taking photos, and people have been doing this since the 50s, basically taking photos from different angles, being able to interpolate information about and measurements. If you know what the scale of your photo is, you can make measurements and also build 3D models off of it. And I know you guys have access to Pix4D, which is one of the many software packages that are doing this sort of photogrammetric processing. LiDAR also has been around for a while. It's light detection and ranging. So it's using this laser or maybe multiple lasers to build a 3D model of the ground. It used to be really crazy expensive because it had to be a particular airplane that flew over, and there were only a couple of airplanes in the US that were even accessible, so you have to pay for them to fly there, and then for them to fly your path. Also, you know that airplanes are much more limited in terms of when they can fly than, say, a drone is, and so you'd also have to wait for that weather window, and did that coincide with when the equipment was available. Now they're making LiDAR small enough that you can use it on a drone. It still generally isn't so great if you need to penetrate through water, but they are improving even that. Still, you're probably only going to be able to take it so far into pretty shallow water or pretty clear water. One thing to think about when you're using LiDAR, though, is what you're asking for and what data are you getting out of it. And in the context of LiDAR, when you think of the resolution, it's how many points are you taking and how closely spaced are there. When you think of the accuracy, it's, okay, well, how close is the value you get for that height close to the actual height? And it's much more difficult to get really accurate heights than it is to get really close spacing. Okay, so that is all the stuff that I wanted to talk about for lecture today. But I want to make sure that you guys are all prepared for our activity today. Emily has printed out for you